Okay, so for Lab 10, this corresponds with Chapter 18, or Chapter 28, the reproductive system. So you'll do this, I guess, next week or so. But I highly recommend you referring to this Chapter 28, 28.3, and 28.4 are gonna have a lot of the structures that you need to identify. There's some great images in there. So definitely utilizing your book for this one will be helpful. And an APR in the reproductive module. They have a lot of different topics. They have female breast and pelvis, male and female perineum, male pelvis, as well as penis, scrotum, and the testes, traumatic cord isolated. So a lot of really good views for the structures we're looking at. Would help if I- Okay, so starting with the female reproductive system, we look at the ovary and the ovaries. So the ovaries are the primary reproductive organ in the female. Let's go to the pelvis. So the pelvis, you have both sagittal and superior views. I'm going to start with the superior because we can get a direct look at both ovaries. So these are going to be producing estrogen. They're going to be responsible for producing the oocytes and developing the oocytes that are ovulated each month that become fertilized. So they sit right there in the abdominal cavity. If you notice that it says under the structure, it talks about the different follicles. So again, if we go to the book, we have to scroll down right here to this image 28.5, this image of the ovary. We also have one of these. So inside of the ovary, there are these different structures. There are these different types of follicles. And a follicle is both the oocyte, it's the egg that's developing inside of it, but it's also the cells that support the egg. So when we talk about a follicle, we talk about the different stages, the primordial, primary, secondary, antral, and mature. The follicle refers to the oocyte and the egg. It goes through those different stages. It starts off as a primordial follicle with a primary oocyte. Okay? And it stays a primary oocyte until it's in a mature follicle. The mature follicle is the only one that contains a secondary oocyte. And a lot of this information that I'm saying is also going to be, now that you have to fill out these questions for whoever's in the class. If you're watching this video, you might have to fill out these pages. But for page like 135, 36, 37, a lot of the stuff that I'm saying is going to answer those questions, if you're interested. But the follicles go through different stages. Starts off as a primordial follicle. About 20 of those are activated to mature each month. So every month, you could potentially have 20 oocytes. You don't. That's not going to happen. It's not even potentially. But there are 20 primordial follicles that are stimulated to develop each month. That fades off. Only one will mature each month from each ovary. But it goes through these stages. So you start off with the primordial, and then it gets into primary follicles. And as it goes, you see that the layers of support cells are starting to change. You start to get this antral fluid. And the oocyte, the egg inside, is changing as well. It's going through stages of meiosis to be prepared for ovulation and fertilization. So as the month progresses, that follicle develops, it becomes a mature follicle, and you notice that it starts to bulge here up against the ovary. So we were talking yesterday, we had a really fun conversation in yesterday's class about one of the women was on fertility treatments for her pregnancy, and so every month she was quite fertile. And so she would say that she could actually feel when she would ovulate because it was just such an intense process, and it would be this like relief. She's like, oh, finally. It's like, you know, because it literally bursts through the ovary when the egg ovulates. All that ovulates is the oocyte, these support cells. Some of the support cells become layers around the oocyte, but then the rest of it will remain within the ovary. This happens every month. That cycle happens every month. Once the oocyte is ovulated, it goes down the tube into the uterus, and two things can either happen. It either gets fertilized or it doesn't. And let's start with it not being fertilized because that's much more straightforward. So the oocyte is ruptured, there's ovulated. Out of the ovary, it goes down the fallopian tube, it doesn't get fertilized. Okay, no big deal. these follicle cells, no matter what happens, these follicle cells that are remaining are going to start to turn into what's called the corpus luteum. And the corpus luteum is there to help support the pregnancy. So whether or not there's fertilization, the corpus luteum hangs out for about 2 weeks after ovulation, and it continues to release hormones to support the uterus. So it helps to keep the uterine lining thick. And that allows for, again, it's just for the first 2 weeks that it's going to start doing that. And that allows time for the oocyte to be fertilized, implant into the uterus. If there is no fertilization, if it's just a normal month, then you ovulate, and then it's not fertilized. After 2 weeks, the corpus luteum will regress into a corpus albicans. This is just essentially just kind of regressive tissue. It's not functional at all. And eventually it is regressed and reabsorbed by the ovary. The corpus albicans is eventually regressed and absorbed by the ovary. So that's kind of the basic stage for follicles. You start off with primordial ones. It goes through maturation to a mature follicle. Once it's ovulated, you get this corpus luteum and the albicans. So I said the corpus luteum is going to be secreting progesterone to keep the uterine lining thick. If there's no fertilization of the oocyte after 2 weeks, this will regress into the corpus albicans. The uterus loses that hormonal stimulus, and so you shed the uterine lining and you have your menstrual cycle. That's what happens if there is no pregnancy, if there is no fertilization. If fertilization does occur of the oocyte, same thing happens. For about 2 weeks, the corpus luteum is going to be secreting hormones to keep the uterine lining thick. During those 2 weeks, the oocyte will become fertilized. It will travel down the fallopian tube and it will implant into the uterine lining. Implantation in the uterus usually takes about a week to 9 days after fertilization. So within that 2 week period that the corpus luteum is still nice and robust producing progesterone, the oocyte can be fertilized, implant into the uterus, and it will start to secrete a hormone. Does anybody know what hormone it starts to secrete? HCG. So the fertilized oocyte that's been, at that point it would be called an embryo because it's been fertilized oocyte embedded into the uterine lining, so we can call it a conceptus or an embryo if you want to. It will start to release human chorionic gonadotropin, HCG, that's what you detect on pregnancy tests. And here's how this little cycle works. HCG from the fertilized oocyte keeps the corpus luteum alive. The corpus luteum secretes hormones that keep the uterus alive, the uterine lining thick and alive, and the uterine lining keeps the oocyte alive. So it has this cycle of oocyte releasing HCG, corpus to support the corpus luteum, the luteum releasing progesterone to keep the uterus lining thick, the uterine lining providing all of those rich blood vessels and nutrients for the oocyte. And the corpus luteum will be what maintains the pregnancy for the first trimester. The placenta does not take over and fully form until about 3 months in. So the corpus luteum is doing that heavy lifting for the first trimester or so.
It stays in the ovary the whole time?
Stays in the ovary. Yep, this is just tissue that stays in the ovary. So yes, that's that basic kind of breakdown on that first one. We talked about primordial, primary, secondary, the luteum, and the albicans. It's all those. And if you have kind of these boards right here, you can see kind of like these white ones are the albicans. This big yellow is the corpus luteum. Over here, you've got like those little popcorn things. I think that kind of looks to me like the primordial follicles, I think. And then you have these over here. You can kind of start to, if you look at what distinguishes primary from antral, a lot of times is the layers of cells. And antral is when you start to see this fluid forming. So that's kind of what determines primary, secondary, antral. Mature are the ones that are ovulating. So ones like this one right here, this would be a mature follicle because it's got an ovulating oocyte. Most likely I won't ask you about a specific follicle stage. What I will ask you, and you can almost be guaranteed, is for you to identify.
The oocyte doesn't change size, it's everything around it is increasing in size?
Say it again?
It looks like on the graph that the oocyte doesn't change size, everything else changes size?
That's a great point. So yes, basically. But real quick, the ovaries, that one you definitely will see. So be able to identify the ovaries in the body. But what you were saying about the oocyte, that's a really interesting point. It does not change size. So as it's developing in here, it's going through meiosis, so it's turning into that haploid versus diploid, so that can be prepared for fertilization. If that doesn't ring a bell yet, don't worry, labs 11 and 12, chapter 28, 29 is all about genetics and inheritance. You'll get to remember all your diploid and haploid. But with the ovulated secondary oocyte, and actually, do you mind if I hold off on your point about that? Just don't, I'm gonna talk about it when we get to the uterine tubes, because that's when we talk about fertilization. So don't let me forget to mention that, if I do. But I'm gonna skip over actually, and we're gonna jump into the ligaments that support the uterus. So the uterus is sitting here in the abdominal cavity. It is not just kind of floating around though. It has these ligaments that help to anchor it in place. The first one that we can look at is the broad ligament. It just kind of covers the span of the uterus. So it goes all the way over the top. It's broad because it is not very specific. I don't have a number on here that I could say, oh, look at this number. It's just this tissue that's covering over the top of the uterus, off to the sides. It's helping to maintain its structure. That's your broad ligament. Yeah, the suspensory ligament right here is this most superior one. If you're looking at kind of these models, it's behind. So with these models, this is the pubis. So this is the front. Here's the rectum in the back. So this is anterior and posterior. The suspensory ligament will be the most posterior. It's those right there. I'm going to show you. It's that one. Yeah. It goes to that one. You can see. Yeah. Posterior. That's the suspensory ligament. The other two, the ovarian and the round. The round ligament is the most anterior. If you're looking at these models, it's the blue one. It's the one that looks kind of blue. It's number 14 if you look in your lab manual. I think this model is on page 133. So the round ligament is number 14. The last one is the ovarian ligament. I don't know. I thought it was. The ovarian ligament essentially is what anchors the ovary to the uterus. So you can see it over here. There's the ovarian ligament and the suspensory ligament. If we scroll, oh, actually here's a good one. So this is image 28.4. Again, your book has great images for this lab. The broad ligament you can see is just covering everything. The ovarian ligament, what we were just going to, that's the one that anchors the ovary to the uterus. It is not the fallopian tube. It's not the uterine tube. It does not transport the oocyte from the ovary to the uterus. It is a ligament that is just holding the ovary in place, but it is attached to the uterus. So when you look at your ovary, there are two tubes that come off of it. There are two kind of tubes right beside each other attached to it. Yeah. The one that is directly on the ovary, that is the ovarian ligament. It's number 20, or is that number, yeah, it's number 13 on the model. It's number 13. The fallopian tube, the uterine tube, is number 10. It's the one that's attached, that's covering and kind of goes in front of the ovary. So can y'all see those two differences? Yeah. Ovarian ligament is not the transport for the oocyte. Okay. Questions about your ligaments? Pretty straightforward. Suspensory is superior. Round ligament is that anterior one. The broad is broad. All right, so now we can get into the uterine tubes. And you see it here. You see kind of this tube. We would traditionally call them fallopian tubes. Now we call them the uterine tubes. There's another image. Lots of charts in this chapter. Here we go. So 28.9. We're going to spend a lot of time in this image. It breaks down the 4 parts of the uterine tube. So we can also see it right here. There you go. Again, the uterine tube, this is what's going to be responsible for transporting the ovulated oocyte into the uterus. Okay? It has 4 different parts to it. The first part is the infundibulum. Okay? The infundibulum contains the fimbriae. That's those little finger-like projections. So those don't actually touch the ovary. They get, during ovulation, right around ovulation, they get really, really close to it. But they basically wait for the ovary to be, the egg to be ovulated, and they kind of catch it and they scoop it up and draw it in. So you have the, showing this. So you have the infundibulum is this very kind of first, like the head of the tube with those little finger-like fimbriae right over the ovary. The second part of this uterine tube, the second part of the uterine tube is called the ampulla. So that's this kind of curved portion of it. This is typically where fertilization occurs. So the sperm are going to swim all the way up the vagina, through the uterus, up the uterine tube, and into the ampulla, and that's where they encounter the oocyte. I feel like it's always a longer journey than people think. So here's where we can kind of talk about your comment, Christopher, about the oocytes while they're in the ovary not really growing in size. So they don't. The oocytes are not going to increase in size until they get into the uterus, once it's like actually the embryo is growing. What happens is that the egg is fertilized in the ampulla. It's supposed to be. It's typically, somewhere in the uterine tube. Usually it's the ampulla. And as it travels down the uterine tube, it's going through a series of divisions. So as soon as the sperm and egg come together and it's fertilized, we call it a zygote, and it's a single cell. So we start as those single-celled organisms. And as it travels down the tube, that one cell turns into 2 cells, 4 cells, 8, 16, 32. But each of those divisions doesn't increase the size. And I always describe this as the difference, if you remember, between hypertrophy and hyperplasia. So if you go and you work out and you lift weights, you're going to hypertrophy your muscles. Your muscles will actually get bigger. If you go through, if you have like cancer and you have a tumor, it's going to increase the number of cells. That would be hyperplasia, right? So in this case, we have this hyperplastic state where our cells are increasing the zygote is increasing the number of cells, but not its size. It's just kind of turning into this hyperplastic thing that is just duplicating. So as it travels through the uterine tube, because the uterine tube is a specific size and is not meant to expand, it's really helpful because you have this development that's occurring within the fertilized, we call it a conceptus at this point, as it's in the fallopian tube, we call it a conceptus. As those cells are replicating, they're actually growing. It's not until they implant into the endometrium and it can start to grow in size. So that's to your question. Yeah, even within the fallopian tube after fertilization, it doesn't start to grow in size until well into implantation in that embryologic fetal period. There are instances where the conceptus will implant outside of the uterus in the fallopian tube. They call that an ectopic pregnancy, ECTOPIC, an ectopic pregnancy. Those are non-viable. There isn't the technology to extract them from the fallopian tube and then reimplant it into the uterus. We don't have that technology, so it's a non-viable conceptus. And it can be fatal to the mother. If it's not removed, the fallopian tube can explode and rupture, and it can be fatal to the mother. Yeah. So that's the 2 parts that we've done so far, the infundibulum and the ampulla. The next part is the longest one, the isthmus. So it's just this kind of long stretch of tube. And then you have the uterine part, where the uterus and the fallopian tube, the uterine tube meet. So pretty straightforward. The infundibulum with the fimbriae at the very front, or the very first portion, the ampulla, where fertilization happens, and the isthmus, where you see a lot of those divisions happening in the cell, going from that zygote to a blastocyst. And then our last little uterine part. One thing I didn't mention about the ovaries with the oocytes, they typically take turns alternating, so left and right. Some of them will say, oh, you don't know necessarily, but it's like the left one's more active or the right one sometimes will become more active. With twins, I think it's always interesting to kind of just mention twins briefly. So the two different types of twins, you have your identical twins. Identical twins occur when one egg is ovulated and that egg gets fertilized. And then through these series of divisions, instead of staying as one cohesive cell with more dividing inside, it actually splits into 2 different cells. And that's how you get identical twins. that is totally random. There is no genetic predisposition for identical twins. For fraternal twins, that's when you have your ovaries release multiple eggs. So for twins, triplets, things of that are not identical, that's when the ovaries will just be releasing more eggs and you have multiple eggs fertilized. And so that's why they look different, because they have completely different, not completely different genetic material, but it's all, I mean, technically, yeah, they have to an extent, 50% different genetic material versus twins, which are going to be on the same.
So that last piece there is called the uterine part because it's all inside the uterus?
Yeah, it's the part that actually attaches to the uterus.
So for the fraternal twins, is there a genetic aspect to that?
Yes, so you are more prone, some people are more prone to releasing multiple eggs at once, in 1 month. Okay, cool. Any questions about the uterine tube, the fallopian tube, however you want to call it? I'm fine with either. All right, let's get into the uterus then. So, the uterus. It is this nice big expandable pouch that can hold the developing embryo, developing fetus. So that first stage after fertilization, we call it a conceptus. Then it takes about 2 weeks for it to implant, and then it becomes an embryo from weeks 3 to 8, and then weeks 9 till delivery, we call it a fetus. So just to keep you, when I use those words, you know where I am. So the uterus is going to hold the developing fetus. Typically, by the way, it's the embryological stage is where you have a lot of foundational growth. I know you haven't done chapter 28 yet, so just to give you that idea, embryological stage is where you're gonna have a lot of your organ systems created. The fetal stage is where growth occurs. That's where things just get bigger, basically. So, the uterus. We can look at there. We can look at it from a sagittal view. The sagittal view is really good. You can see the uterus right in here. You have the bladder in front of it and the rectum behind it. Okay. We could look at the lumen, the fundus, the body, and the cervix. So 4 parts of the uterus: the lumen, the fundus, the body, and the cervix. The lumen is this inner portion. That's the portion where the fetus and the embryo are actually developing and growing. I don't have a highlight for that. It's just the inner portion. Oh, here you go. Yeah, 28.9, that picture. There is showing, kind of pointing to the inner portion of the uterus. We should know what lumens are by now. We know a lot of lumens. Next, you have the fundus. So the fundus, if you remember, hopefully you got this correct on your lab practical, we had the fundus of the stomach as one of the answers. So the fundus is this kind of rounded upper portion of the uterus right here. If you're looking at the model, there's no number for it, but it's essentially just kind of this rounded upper portion of the uterus. So the very kind of superior region. That's the fundus of the uterus. The body, again, that's what you think of as the body. It's the main section of it, right, the main portion. And the last bit, the cervix. The cervix refers to this general region. It's kind of the entryway to the uterus. It's between the vagina, that tube, and the actual lumen of the uterus. The cervix has 3 regions: an internal and external os with a canal between. The external os is between the cervix and the vagina, so it's outside. The internal os is between the cervix and the lumen of the uterus. So it's inside, it's deeper. So just to keep those two straight, you have the internal os deeper in the uterus, the external os closer to the exit. And os just refers to a hole. It just refers to a space or a hole. And then the space between is the cervical canal. So that little space between is the cervical canal.
So it only goes between those two, it's not the whole length?
It's not the whole thing.
Okay.
Yeah, it's just from this to this. So when you're looking at these models, and I'll definitely come around and show y'all, you have this kind of big Y-shaped cup thing. That's referring to the cervix. So that opening just before it goes into the uterus, that's the external os. And then you see it has those squiggly little lines on it. Just where those squiggly little lines end, that's the internal os. So internal os, external os. External, and then internal. seen is this canal. Okay. Questions about your cervix?
No, but on the model, it's more horizontal, and it's a little more, with the graph's a little bit more.
Oh, well, it's always going to be different in the body.
Oh, is it?
Yeah,
Okay.
yeah. Especially with a good outer model. Yeah. The layers of the uterus. Okay, so the uterus has 3 layers. This should be pretty familiar to us, this naming convention. You have the innermost layer, the endometrium, the middle layer, that muscle myometrial layer, and the very outer parametrium. So the outer parametrium is actually continuous with the broad ligament. That is the same thing. The parametrium and the broad ligament are just one continuous structure. The myometrium is this thick muscle layer. Why does the uterus need a thick muscle layer? Contractions. Yeah, it's got to get that baby out of there. The endometrium is that inner layer. You can kind of see it on the model how there's a transition between the muscle and another layer of cells, most internal, kind of closest to the lumen. That's the endometrial layer. That's what is shed every month. It will develop and get thick right here. It's nice and it's kind of small and thin. At the peak, it will be almost as thick as the myometrial layer. It gets to be very, very vascularized, and then every month when there's no fertilization, it gets shed out as well. Yeah. All right. Any questions about your uterus or someone else's? if you don't currently possess one. All right. The vagina going into the tube. This tube that exits from the uterus out to the body, outside of the body. That's pretty much all it is. It's a muscular tube. Yeah, you have your urethra, vagina, rectum. We can look at it from the perineal view. There's the vaginal orifice. That's one of the things that you need to know. Just anterior to it is the urethral orifice. I thought the vagina is pretty straightforward. Next, we can do the external genitalia. So the mons pubis is this region of tissue in the very front, kind of lower anterior abdominal region. Looking at your model, is that little bit of fat right in front. Right there. That's the mons pubis. These models we were using don't show it.
Lack that.
Yeah. The others, the labia majora and the labia minora. So those on the model are the labium majus and labium minus. So one set's large, one set's smaller.
Would that be considered inferior, superior, or just smaller?
Not. That would, if anything, I would say these are deeper and these are more superficial.
Okay.
It's either more medial and lateral. You got the minor and the major.
Okay.
Those are going to be analogous to the scrotum in the male. They're going to help provide protection. The clitoris, we're actually going to shift to the perineum view, and we can see the body of the clitoris and the actual whole structure. The external urethral orifice, this is repro, so we don't have that marked, but you should still be able to know where it is. The vaginal orifice, we did that one. The bulb of the vestibule and the greater vestibular glands. You have the greater vestibular glands and the bulb of the vestibule. These will secrete. Any questions about the external genitalia? Pretty straightforward. Last are the breasts, so we can move to the female breast tissue. The nipple is the actual projection off of the breast. The areola is the darkened skin that's around it. After pregnancy, postpartum, that typically becomes darker. And we were talking yesterday, and one of the women was saying, oh yeah, like they've shown that it actually makes it easier for the baby, the infant, to see the nipple and the breast. So they can kind of zero in on it a little bit more. Because at that point, they really don't have great depth, like far away vision. So it's right there in their face, like there's something dark there, I'm gonna go for that. And they do, with gusto. All right, and then the lactiferous ducts. So these are the milk ducts. We can go into the very bottom of this chapter. Yeah, figure 28.13. And you can see in here, because your lab manual mentions the ducts and the sinuses. So we can see that as the ducts, the milk is produced within these alveoli. We have alveoli in our lungs, we have alveoli in the mammary glands. Milk is produced here. It's collected in the lactiferous ducts. And then when the ducts join together, they become these larger sinuses. That's just what the sinus is. Lactiferous sinus is just when the ducts join together, and the sinuses will exit out through the nipple. Notice it's not just one hole out to the nipple, it is multiple ones. All right, any questions about the female system? That was pretty much everything. All right, then let's jump into the male system. So with the male system, I like these models. See if I'm gonna have a little bit of everything. Jumping over to the male system. With the male, again, we have the pelvic view, but then we also have this penis and scrotum isolated. So we're going to start here. So in the male reproductive system, the first structure is the scrotum. The scrotum refers to the sac of skin that that contains the testicles. See it there. You can see it there. Inside of the scrotum, as we said, are the testes, the testicles. You can see them isolated in this one as well. That's a good. The testicle is analogous to the ovary in the female. It's going to produce testosterone, make sperm. It will produce and mature them, and then they are stored in this structure outside of the testicle until they can be ejaculated out. If we look inside of the testicle, it's divided into these different lobules. So you have these lobules, the septum dividing the testicle. We're actually going to go to 28.4 and scroll down to figure 28.16. So in figure 28.16, you can see again we have the testicle inside of the scrotum. Within the testicle, it's not just this hollow tube, it's divided into all of these partitions. It's divided into these lobules by the septum. And inside of those lobules are what's called the seminiferous tubules. Okay, so this is one of the first structures. And the testes is the seminiferous tubules. This is where sperm production occurs. So if you look at this histology image, you can see that out here towards the outside, what happens is that they start out here, and then as they move through the tubule system, the sperm mature, they become more and more mature until they enter the ret testes, and then are stored in the epididymis. But if you look at this section of the tubule, you can see kind of towards the periphery, they start off as just kind of generic looking cells, but as they start to move deeper, they're transforming, and they're turning into sperm. So you start off with spermatogonia, these immature sperm cells. As you move deeper, once you can kind of see the nucleus condensing, they become spermatids. And then if you look here, you can actually notice, if you kind of look carefully, it looks like squiggles at first, but you actually see the head and the tail in that classic sperm shape. So here are the sperm. So just like the ovary has to go, the oocyte goes through meiosis to turn into a secondary oocyte, same thing happening with the sperm. They're going from these diploid spermatogonia cells, which always stay the same. They always kind of keep the constant number, constantly producing sperm, unlike oocytes. So males produce sperm into their 70s and 80s, no problems, lucky them. So the spermatogonia cells, they never really decrease in number, but they keep producing these spermatids, which turn into sperm. For every one spermatogonia that matures, you get 4 sperm. For oocytes, it's the opposite. You get 1. You get 1 oocyte. So that's just kind of those structures that you need to know real quick. The seminiferous tubules, where the sperm actually produced within the ret testes. They then are transferred through the efferent ductule. You see this little piece of tubing between the ret testes and the epididymis, that efferent ductule, before the mature sperm are deposited within the epididymis to mature. So yeah, just the seminiferous tubules, ret testes, efferent ductules, spermatogonia, spermatids, and sperm. These sustentacular cells, these Sertoli cells, those are support cells for the developing sperm. That's what we're doing. Yeah. Cool. The epididymis, that structure I've been mentioning is surrounding the testicle. It's within the scrotum, but outside of the testicle. Okay? And you can see how the tubule system works, is how it connects to the epididymis through these efferent ductules, and it's going to store the sperm within the epididymis until it's time for ejaculation. And you can see that as you travel kind of down through here, you get to the tail of the epididymis, you start to come up, and it goes up and out. So this tube only goes up and out. It only travels one way out of the body. You can see the epididymis there, surrounding the testicle. So again, outside of the testicle, but within the scrotum. This cord right above it is called the spermatic cord, and I'm gonna have to. Here you go. So the spermatic cord. So this is what it looks like wrapped in tissue, wrapped in connective tissue. The other version we saw was it not in that connective tissue, so I'm going to switch back to that one, because the cord itself, but this way you can see this stuff. The cord itself is made up of multiple structures. You have primarily the ductus deferens, or the vas deferens, and then you have the testicular arteries and veins. So these are the testicular arteries and veins, as well as some of the muscles that help to control movement of the testicles, and then the ductus deferens. So the spermatic cord just contains all of those things, containing all of those structures. The arteries and veins, that's pretty straightforward. The ductus deferens, or the vas deferens, that is this tube connecting from the epididymis that is going to insert into the urethra, and it's going to be a transport for sperm. So when it's time for ejaculation, they leave the epididymis, they travel up the ductus deferens, and are ejaculated out. This is similar to the uterine tube, the fallopian tube within the female, as it's the transport for the gamete. Just like in the procedures for these two are the same. For a vasectomy and for a tubal ligation, they're both cutting this cord. So in the male, for a vasectomy, you cut the vas deferens, you cut the ductus deferens, and that just blocks the sperm, their route to be able to get out of the testicles. With the female, you can ligate or put a clip on the fallopian tubes, so that way the oocyte can't get out and the sperm can't get in. At that point, things are just regressed and the body reabsorbs them.
Oh.
Yeah, was that gonna be your
Yeah,
question?
no,
It's like, what happens?
Yeah.
Yeah, the body reabsorbs it, it's okay. Yeah. I will say on this model, you have this image of the male repro in your lab manual. You can kind of see in the testicles, it has some kind of remnants. I think it's trying to show what these tubule systems look like. So you've kind of got like the seminiferous tubules kind of right in there in the bulk of the testicles. And then you can kind of see this almost like parrot looking beak that curves over top of it. That's supposed to be the epididymis. Is that little part that's kind of curling over top. So still in the scrotum, but kind of outside of the testicle. That's my best interpretation of this model. Oh, shoot. Oh, things off.
It comes up. I was like, I don't understand. And then I looked up and it was.
You have found what every 4-year-old boy finds fun too. So, spermatochord, the ductus deferens. So yeah, as we said, the ductus deferens, you have the epididymis. The ductus deferens is going to be leading out of that. It goes up and around and behind, and it inserts through the ejaculatory duct into the prostatic urethra. Okay, so the ductus deferens coming up, around, it passes above the seminal vesicles. Right, so that's the seminal vesicle, passes around that, and then right through that little duct, that's the ejaculatory duct. That little line right there is the ejaculatory duct within the prostate. And again, that's where the vas deferens and the seminal vesicles are going to be depositing their contents. So the vas deferens has the sperm that it will be depositing. The seminal vesicles, they help to produce fluid. The seminal vesicles, the bulbourethral glands, the prostate gland, they're all producing fluid to help support and nourish the sperm, protect the sperm as they go on their journey. So that's what these kind of things are going to be. So yep, there's that. Let's see. Yeah. There you can see just posterior to the bladder is the seminal vesicle. It's not a great image of it, but it's there. This right here is your prostate. Right there below the bladder. Seminal vesicles, ejaculatory duct, prostate gland. The bulbourethral Cowper's glands, you can't see it. They don't have it on the APR models. I'm pretty sure they don't. I can't imagine where they would. Yeah. So we're gonna go to here. Oh, there's that process of spermatogenesis. Sperm being made. So again, you see you have these one cell in the periphery, and then as it moves deeper into that lumen of this seminiferous tubules, they actually become sperm. Here we go. A little bit. Sorry, I'm trying to find. Oh, at the top. So this very first image. There's your bulbourethral gland. So you can see just beneath this membranous urethra, once we actually get into the shaft of the penis, then you have this bulbourethral. There's a pair of them, shown on the other side, that are going to be releasing into the urethra as well. So figure 28.14, that's going to be really helpful. You get to see the ductus deferens, you get to see the seminal vesicles. And notice here, I skipped over this one, the ampulla of the ductus deferens. The ampulla just refers to kind of the swollen portion, right where it's joining here with the seminal vesicles and forming the ejaculatory duct. So that's that last bit is the ampulla of the ductus deferens.
Okay, so that's why it's underneath the ductus inverted.
Yeah.
Okay.
Because it's a part of it. Okay, any questions for that stuff then? We covered and did epididymis to the bulbourethral Cowper's glands. I know I kind of breezed through the prostate, but again, it was male pelvis. That's a good way to see it right there. Okay, moving into the penis then, we can talk about the different portions. There is the glands of the penis, the head of it. That is analogous to the clitoris in the female. The prepuce. And we'll have to go to this one. Yes. So in this model, it's a circumcised male, so it's just the remnant of the prepuce of the foreskin. Otherwise, the glands would be totally covered by that. And just, there's the glands. We could look at the 3 different regions of the urethra. We've done this already, but just to kind of refresh, you have the membranous urethra just below the prostate. You have the prostatic urethra, which is running through the prostate itself. The ejaculatory duct inserts into the prostatic urethra. That's the region of the urethra that it inserts into, is the prostatic. The final portion is the spongy urethra. Hopefully you got this correct on your practical as well. That's what that one was, is a spongy urethra. It's called a spongy urethra because it runs through the corpus spongiosum. So within the penis, there are three erectile bodies, we call them. The corpus spongiosum is one that runs the entire length of the penis from the base all the way to the glands. The cavernosum only runs up until the glands. It stops before the glands. And there's two of these kind of side by side. So here's one view of it. And then, but again, these are the erectile tissues. This is what actually is allowing the penis to become erect. So you can see, yeah, they're running the length of it. If we look at it this way, you can see the cavernosum, so the two layers of the cavernosum stopping just before the glands, and then the one spongiosum that runs the entire length.
Oh, so that's why you say there's three, there's two.
Yeah, three bodies, because it's 1, 2, these are divided by that septum. Yeah. Questions about the penis? Any of its structures? Cool. Last two things are the dartos and cremaster muscles. Again, they're not here on these models, unfortunately. We just don't have good ones yet. But it does show up in this image. So 28.15. You can see the cremaster muscle is part of the spermatic cord. So it's helping to raise and lower the testicles, because sperm production requires them to be slightly cooler than body temperature. So the testicles will raise if they get cold, or they'll drop lower if they get hot to adjust temperature. That's through the cremaster muscle. The dartos muscle is within a layer of the scrotum, and it can help to tighten or relax the actual skin of the scrotum to, again, regulate temperature. Okay. Any questions? No?
So not all of them will be in APR then?
No.